This is an overview of an article entitled Whole-Exome Sequencing Identifies Pathogenic Variants in TJP1 Gene Associated with Arrhythmogenic Cardiomyopathy, published in the October 2018 issue of Circulation, Genomic and Precision Medicine. Arrhythmogenic cardiomyopathy, or ACM, is a common cause of sudden cardiac death in the young, and, in particular, the young athlete. ACM is defined by replacement of the normal cardiac myocardium with fatty or fibrofatty tissue. The subform, predominantly involving the right ventricle, is the most common, but left and biventricular forms have also been documented. Myopathic changes are associated with increased predisposition to ventricular tachycardias. Believed to be genetic in etiology, approximately 60% of individuals with ACM are mutation positive and most often host mutations in genes encoding components of the cardiac intercalated disc. These include PKP2 encoded plakophilin 2 and DSP encoded desmoplakin, among other proteins. Mutations in non-intercalated disc genes have also been associated with ACM and are rare. While so-called mutation-positive individuals have a known genetic etiology of their disease, approximately 40% of individuals with ACM have no known genetic cause for their disease and are so-called mutation-negative. Tight junction protein 1 is encoded by the gene TJP1 and is also known as zonula occludens 1 or ZO1. As its name implies, it belongs to a family of proteins that forms a scaffold which crosslinks and anchors other components of cell-cell junctions. In particular, in the heart, the protein is essential for normal organization of gap junctions, which directly connect the cytoplasm of two cells, allowing ions and electrical impulses to directly pass through a regulated gate between cells. Mutations in TJP1 have not previously been associated with ACM or any form of cardiomyopathy. In the paper under discussion, the authors sought to determine whether variants in TJP1 are associated with genotype-negative ACM. The authors had performed whole exome sequencing on a proband who demonstrated ACM but did not have a mutation in a traditional ACM-associated gene. Following identification of a rare variant in TJP1, the authors searched for additional TJP1 variants in a cohort of 40 unrelated Italian probands with ACM who were similarly genotype negative. The authors then sought to validate their findings of TJP1 variants in a second confirmatory cohort of 43 Dutch-German patients previously sequenced by whole exome sequencing. Finally, to attempt to determine whether identified variants were functionally impactful on TJP1 protein, the authors performed in silico modeling of protein stability, which was tested by molecular dynamics simulations and predictions of local structure content. Following genetic analysis in genotype-negative ACM individuals, the authors identified 4 missense variants in TJP1. The Y669C variant was found in the proband of the first family, was predicted to be damaging by 8 in silico tools, and localizes to a highly conserved amino acid at the guanylate kinase-like, or GUK, domain of the protein. Targeted genetic screening of TJP1 in 40 Italian ACM patients identified a second missense mutation, R265W. This variant was also predicted to be damaging by multiple in silico pathogenicity prediction tools. Analysis of whole exome sequencing data from an independent cohort of 43 Dutch or German patients identified 2 additional TJP1 variants, S329L and D360V, in unrelated probands. The S329L variant was found in the NOMAD database with a frequency of 0.006% and is predicted to be damaging by 6 in silico tools. The D360V variant, identified in a proband who also hosted a pathogenic variant in the DSP gene, was predicted to be damaging by 5 in silico tools. The initial proband and his family, who were found to host the first identified TJP1 variant, is shown here as Family A. In the pedigree of Family A, the missense mutation Y669C co-segregates with ACM. In the pedigree shown here, individuals hosting the Y669C mutation are noted by a dot, and the affected family members are designated with standard black fill. A second pedigree for family B, to whom the second described proband belongs, is shown at the bottom of the figure. In this pedigree, the missense mutation R265W co-segregates with ACM. To provide evidence that the predicted pathologic variants impact the functionality of the protein, the authors identified that R265W affects a highly conserved region of the protein, as shown in panel A. Using in silico modeling of local protein structure, the R265W mutation was predicted to destabilize a PDZ2 beta strand, as shown in the lower portion of panel A. To explore the impact of the Y669C mutation, which occurs in the GUK functional domain of the protein, as noted earlier, the authors performed molecular dynamics simulations, as shown in panel B. 2 independent runs of molecular dynamics simulation showed a more open conformation of the GUK domain in the mutated protein. Analysis of the trajectories showed the wild-type protein to be relatively more stable than the mutant. The authors concluded that the Y669C mutation likely is associated with local unfolding of the GUK domain. Taken together, this study made 3 main observations. Pathogenic variants in TJP1 were identified in 2 families with genotype-negative ACM and in 2 Dutch-German patients. Thus, rare variants were found in 2 independent cohorts of ACM probands, linking TJP1 variants to ACM for the first time. In silico modeling of TJP1 mutations supported a hypothesis of functional protein impairment through structural instability and loss of appropriate protein function. In conclusion, while ACM is predominantly associated with impairment of the mechanical functional unit of the cardiac intercalated disc, identification of TJP1 pathogenic variants underlines the potential direct involvement in ACM pathogenesis of the electrical functional unit, composed by gap junctions. ACM-associated variants in TJP1 may impart a risk for ACM through structural rearrangements of the protein. Finally, additional studies are needed to fully explore the role of TJP1 in the heart and the functional consequences of disease-associated variants.